found. Oh wait, yeah, we need to share the slides, or it's going to be a discussion, just like a
Or
pure
I got the slides.
improv. Okay.
Here we
Um,
go.
yeah,
Is the screen sharing? Great.
yes.
Well, hello everyone. Welcome to the Trend Center Machine Learning Barbecue Hurricane Edition, where we're all online. Today, I wanted to go over more methods and applications of work that I've been doing with the Keilholz Lab over the last several years, and give motivation on sort of what these methods are, a little history on sort of how they were used, and sort of what advancements we've been going forward with. Today's a very loose presentation, so feel free to unmute me. I got way too many images from way too many papers, so hopefully it'll be sort of a nice discussion, or if you do have any questions, please let me know. I got my COVID booster shot 2 days ago, so I'm still a little under the weather, so if I'm a little scatterbrained, I apologize, but hopefully that'll help. During the presentation today, I hope to go over some motivation, algorithmic details, since we are very much a CS-oriented group, so knowing a little bit more of the details is fun, but give you applications of QPPs and some complex principal component analysis and a little bit of discussion. Let's talk about some motivation. So as we know, with a lot of fMRI data, we end up getting matrices that end up looking like this, where we have some sort of atlas or neurological structure that we are mapping to over a time. And so we typically see a lot of this where even just visually inspecting, we can see some regions go off and on, and maybe there's some kind of correlation or pattern of networks that are corresponding together. We can take this further by looking sort of at the sliding window correlation and phase synchrony, both of which are older methods to try to make sense of what is potentially happening, both in a spatial orientation or a time orientation. And so one thing that I really like doing this visualization here is, especially with the sliding window correlation, we start to see some patterns emerging, or similar structures just empirically and visually. Same thing with the phase synchrony, which is just the phase at every time point correlated to the matrix. These are more static analysis of the time series matrix, but we can start to see patterns go through it, which offers some motivation to try to find, is there a pattern, underlying pattern or structure to this data that we see with the time series. And so what we end up finding is this QPP algorithm was developed to start to look for this iterative template, or this iterative pattern. Back in 2011, Wakat Majed and Shella Keilholz ended up going through in adding a little piece to the sliding window correlation. And so essentially, this is the visual representation of the QPP algorithm. I have 2 slides to go over more of the finer grained details, but what it is in a nutshell is that there's a template that is iteratively updated over the whole length of the scan or group of the scans, where they will find either a threshold of correlation between that scan piece, and then the template is then averaged and updated throughout the iterative process. This can be done using random seeds throughout the whole group scan, or every initialization point for the group scan as well. Going through, as a template gets updated, there's certain criterion for thresholding, for finding the exact matching of this template. Then they receive the final template. One thing that they were working on in 2011 to 2014 is looking at the optimal window length, and I will hopefully show a reason why that is important here in a second. But the window length is sort of a hard press definition as you try to find the template matching. It is very iterative. It has a complexity time that is linear. So if you have larger scan times, the processing time will be greater. One way that they hopefully found a way to increase the speed of the computation is to find random initial segments and not have to go through the whole algorithm as they've compared over time. The fast or robust or the fast way of computing the algorithm using random segments has shown to be just as good as the full initialization of the robust QPP. But what they have ended up doing what we have ended up doing over time is giving this both in human demonstrations and rodent demonstrations. And hopefully I can show you that that is true over time. Going back to the paper from 2011, understanding that these templates can be very long, they can be very short. These were the initial findings from both rodent and humans. One thing of a question is how long are these waves actually? And they differ by species. One thing that they were finding with rats is that they have a much faster propagation of waves. And what they find is that they typically are around 18 seconds in fidelity, where they have these corresponding waveforms, whereas in humans, it's around 20 to 30 seconds, depending, where they have the most optimal correspondence. As you can see, as you increase the length of your window, they will still continue to be corresponding to each other. However, what is very interesting about the waves is that we see this anti-correlation between the default mode network and task positive networks. And this is very important, as this is a known psychological or physiological and psychological finding from previous studies where these relationships are well known and are theorized to coordinate our brain states as we shift from potentially doing a task or potentially being idle. And so, as you can see, these waves propagate from region over time. And this was a great finding by Wakaz. Further, in 2014, Garth Thompson, who was a postdoc in Keilholz Lab, ended up doing local field infraslow local field potentials corresponding to the bold signal in rodents, where they were able to get a local field potential reading in coordination with the bold signal, where they were able to find these infraslow wave patterns throughout the brain in the rats. So demonstrating not only that these occur in bold signal, but they also occur with local field potentials throughout the whole brain, indicating that there might be some coordination for the whole brain with these ultra slow signals. As the lab sort of progressed and new students started to come, Anzar Abbas ended up using this same type of model, looking at the difference between resting state and task, trying to understand if these signals of anti-correlation differed between task and resting state, where they did find that task did have some implications of the specific regions that were active, mostly visual and somatomotor. But many of these, basically, this paper concluded that there is major differences between task and rest, and various tasks might have different effects depending on what the task might be. Further, because this is an iterative method where we are extracting the QPP patterns from the original signal itself, it begs the question, is it actually picking up the global signal or any kind of respiratory signal, or what are these actual signals that we're picking up from the brain actually doing? And so Banaz Yousafzai in 2018 and 2021 or 2020, I can't remember the year, but she ended up going through and finding the correlation between the global signal and many of these patterns. We have a high fidelity when global signal is not removed from the original scan and pre-processing, that the first QPP picks it up almost in its entirety, 0.999% fidelity. But with this, that is saying that there is some that these waves do propagate and explain away a majority of the variance that we see within the signal itself. And so typically when we do global signal regression or QPP regression, we typically only go 3 levels down, if not only looking at the first 2 waves in itself. And so those are the major findings from her, understanding that the global signal is one of these widespread waves, and that the first QPP does explain a majority of this functional connectivity coordination. Great. As we look to differences, what can we apply these QPPs to? And so Michael Belloy and Abas or Anzer Abas ended up doing comparisons against neurological diseases or known sort of groups that we can test against. And so Anzer in 2019 ended up doing an ADHD comparison between humans where they were able to find the default mode network in the most anti-correlated regions to define the task positive. They were able to compare between the 2 groups using the differences between the magnitude of the QPPs, showing that there is a significant difference between people with ADHD in control, giving us a more lower hanging fruit for a biomarker of ADHD. Since this is a global signal throughout the whole brain, we think that there is some coordination deficit happening within this disease, and QPPs might be a great way to sort of differentiate that. Further, we have Michael Belloy in 2018 looking at diseased mice, specifically with Alzheimer's. And we're using the template from the healthy control in using that to see how they differed with the diseased rodents. What they were able to find is that there is a decrease in visual and somatosensory modes, as well as task positive coordination, specifically with the dorsal attention being very much implicated as an area that this disease does affect. But what this is telling us is these are great biomarkers, especially at a higher level of finding at least differences between controls and disease groups. Great. Okay, great. And even further, more newer work by Nan Zhu and Harry Waters, looking at sort of what happens at different areas of both task and rest. And so Nan came up with a nice little metric where we're looking at the phase shift between these waveforms, as we know that there might be a nice onset and a decrease as these waves do propagate, finding the initial starting point, either if it's going on or off, was part of this new algorithmic coordination, or this new algorithmic detail that Nan included with the QPP detection. But the way that they were able to look and compare against task versus rest is to look at the phase shift and look at the individuals versus group. This was a visual task that they were doing and looking at the difference between sequential videos or a randomized shuffle of visual items as well. And what they were finding is there are stark differences between tasks, and you can measure that with the phase shift of the QPP between rest and task. And so this gives us a nice, a new metric to look at as we compare the way that the the way that the subjects were actually using or actually being tested. To make this even further, this new work is still in preprint, but this is what Harry's been working on extensively over the last year, is to look at more of an axis of attention. And so there are, I believe, 8 or 9 different data sets with different task data here, ranging from external attention to internal attention. There's a visual task, congruence, rest, meditation, mediation, and twilight zone visual, like a visual movie. And so what we can look at then is to look at the QPPs in relation to all networks and not just the task positive, but also dorsal attention, ventral attention, frontal parietal, visual, in in the somatomotor. And we can look at how these tasks differ more so in terms of an axis with a lot more data driven.
I have a
Yeah,
silly question, TJ.
silly
What
answer.
episode of Twilight Zone did they show? Do you know?
You know, that is a good question. I don't think Harry's on right now, but no, you didn't make it on. I believe they're using the Chucky episode, the one that Chucky's based off of.
Oh, I don't think I remember
Um,
that one offhand.
yeah, it's the one where a doll comes to life and only one person can see it. So it's supposed to evoke fear and suspense and like a whole litany of things.
Very cool.
Um, yeah, so Harry's been going through, he has way too much data. He's been cut off of trying to collaborate with new people. But what we've been finding is that there are implications for different networks, really depending. The dorsal attention has the most interesting, at least anti-correlation change between the default mode network. And what we're finding is using correlation between the default node network and the other regions defined here is actually a good method of comparing for groups. And so this gives us both a way at looking at the template of the spatiotemporal patterns, looking at them over time with groups and comparatively against other data sets as well. And so this is not everything that QPPs has been doing recently. Lauren Daly, who's another member of the lab, I didn't have time to put her research in here, but she's also looking at an axis of arousal, where they're defining more internal stimulus, external stimulus, looking at different disease states for narcolepsy and different diseases as that such, but looking at deprecation of attention due to neurological effects rather than task is another method that our lab is going into at this point. Those results are coming out briefly. Lauren just gave a really nice proposal talk about a month ago, and so hopefully that research comes out sooner than later. But this does give some implications and ask questions of, well, if we're doing this as a sliding window correlation, this is very iterative. We have sort of these templates with hard defined they're defined cutoffs for thresholds. Why don't we try more of a data-driven approach rather than doing this more iterative update? And so Taylor Bolt, who was a postdoc with Shella, now works with Thomas Yo and a couple other wonderful researchers. He came up with a method several years ago looking at the complex component or complex principal component analysis, where they basically assume that there is a complex component attached with the fMRI, looking specifically to separate the data into its principal components to be able to discern between some of these potentially global signals or global artifacts that might be propagating. And so what they end up doing is, instead of just going directly on the data, they take a Hilbert transform on the real value bold data. That way we can get both a real and an imaginary part to be able to better deal with the phase shift or the way that these waves do propagate if they're out of sync to better come up with these separations. But going through it, this is pretty standard PCA after doing the Hilbert transform. We got to interpret it. We have amplitude, we have phase, there's phase delay maps. We can retain different end components, but yes.
Now I have more of a real question, and since Ashkan's not here, I'll play the signal processing advocate for him.
You got it.
So the standard SWPC acts as it's a low pass filter, essentially, right? And I imagine the previous method is subject to that, and this would be as well, I guess. Even if you add a complex component, somehow capture phase. I'm a little bit unclear about that, but wouldn't you still be filtering the data by using correlation as your connectivity measure without doing any modulation of the frequency band, which is what Ashkan does in his work.
Correct. So with QPPs specifically, we have already bandpass filtered the data. So this is already within the infraslow range, which was just a pre-processing step. Here, we don't need to do that as much. And Taylor does in his paper show the way that the phase lag, or the using the Hilbert transform with the principal component analysis does account for the phase shift. But yes, it does. I see where it's coming, or see where you're coming from. It
Yeah, I wish
is a
Ashkan
low.
was here because he knows more, of the details, and his paper is really, he has a couple papers talking about how, Pearson correlation works as a filter. Even if you pre-process filter your data, it's still going to filter your data again, right? It's going to filter that bandpass signal.
But filtering it for, I guess, in terms of it's filtering it, how so?
It essentially, if I recall correctly, acts as a low pass filter. So there's for the standard DFNC stuff that we do, it's an issue because we also bandpass.
Yeah, but we're also,
yeah,
I
and
guess
then
we don't need the higher, if it is still bandpass filtering or doing some kind of filtering, the higher frequencies that we're usually seeing in bold signal are going to be respiratory, they're going to be cardiac pulsation, they're going to be sort of breathing even. And so that's not necessarily
potentially, it depends on what range you're looking at. But yeah, I wish Ashkan was here.
Well, yeah, because there's going to be certain bands that have different implications.
It's the windowing that you do with standard SWPC that becomes an issue. And I missed in this method if there's any windowing that happens.
No, so with this one, it is agnostic to window. And specifically, it is looking for the maximum correlation. Yes, I believe it is still using the correlation value, the Pearson correlation value. So I can look into that. That's a good question.
Yeah, just the details of the method, I'd wonder if it has that same problem, but maybe how Pearson correlation is applied here is quite different. And I just
no, you are correct. Let me, after this presentation, Brad, I'll go back and look. The Taylor's paper ends up citing really Facebook's method of the way to do singular value decomposition through this, and it doesn't have the exact details of the method, but I'll go through it and follow up with that.
Yeah, cool. Thanks.
Yeah. But lo and behold, what this ends up doing, especially with non-global signal regress data, is the components do end up corresponding to to quasi-periodic patterns almost identically. What they were able to find is that when you do not use global signal regression, this method will pick it up almost in its entirety, whereas the second component will correspond to the first QPP previously shown with the other methods. Taylor then went through and ran a bunch of different types of organization of brain dynamics mapping. So looking at different caps, looking at hidden Markov models, looking at HGM for different caps, but the correlation of what they're showing is the reason why he's showing this is that the principal component analysis do end up they still explain a majority of the variance within the signal. Looking even at the lag projection of the maps, the different ways of looking at the different phases, they're finding that the components of the quasi-periodic patterns and the principal components do end up matching up and has these similar fidelities.
Sorry to interrupt again, but can we just go back to what the method is doing? Because I'm now confused. Is it doing excuse me, is it doing a Fourier transform and working in that complex space? Is that what we meant? Or is it, what I heard is you're adding a complex component, and that totally, I think, threw me off. But are you doing a Fourier transform then working in the dual space? And that's where you get all this phase information and lag information. That would make a lot more sense.
Yes, sorry if I apologize for not being clear. Yeah, this is taking a Hilbert transform specifically.
Oh, it's a Hilbert transform, right? Okay, I missed that step. Duh.
I
Okay.
apologize. Yeah, so
No,
that's
my
why
bad.
this is not just PCA. This is the complex component happens with the Hilbert transform, so we can get the phase shift.
Got it. Okay, and then you're doing a complex correlation matrix in the dual space. Okay, so the filtering is not going to be a problem in the same way as if you're
Yeah,
getting correlation in the real in the time space, like we do with
correct,
DF and C. Then it acts as a filter.
correct.
So I think my question is answered by me better understanding what's going on here.
Yeah,
Okay.
I apologize. That's on me then. I will clean that up as I explain
Oh
that
no,
for
that's
future
me.
presentations.
I think my attention lapsed for a second there. I
No,
missed-
it's all good. So really what this is doing, since it is splitting up the signal in the complex plane, we get this agnostic phase. This is a toy data set that Taylor was using to show that even with a delayed phase, they're able to still take the or understand all the variance between the signals and basically demonstrate that the components coming out do actually mean something and can split up the signal effectively. And so then applied to the Human Connectome project, they were able to demonstrate that there are really three major components to the or cPCA, and then correlated it back to the QPPs and understanding how those work. So with this being said, we do not have we don't need to define a window length now. This is more of a data-driven approach than an iterative approach. This is new work that I've been working on. This is just a nice little comparison to try to show. But QPPs on the left, cPCA on the right, trying to look at the different waveforms here. I apologize for the sparse graphs or the sparse labels. And this is just messy in general, but looking at the different QPPs, we still see great fidelity. These are polar plots that are correlation values for the default mode network and every other network. So just trying to look at different shape values. There are differences with algorithmic choices, or there's different implications between the algorithms, and then there's different ways we can look at it. Looking at different data sets as well, we still see great fidelity between the two. This is taken at the CABI Center over at Trends, but it also begs the question, and something I've been working on, is how much data do you actually need to have some of these corresponding relationships between the default mode network and the other network. And so this is just some preliminary results from how long group scans need to be versus sort of the mean correlation between the default mode network and these other networks. Here I've also timed as well, since we're taking a Hilbert transform as well as PCA, it is in the cubic complexity or time complexity. And so we should see a slower start and then a longer rise, but I didn't even get there with scan lengths upwards of 20 hours. All this is implying is that cPCA does have a lot more or a lot less variance between between network correlations with the DMN as you increase, and it's a lot faster. But how am I doing on time? I know I've been kind of rambling for a minute, 35 minutes. Okay. But this is a majority of what I had for a discussion. These are still very preliminary, but happy to chit chat about other aspects or applications. Really what I hope to see happen in the, as I finish up my PhD and all this research, hopefully I can apply this to more neuromark results and try to get a little more disease states using this method. But yeah, that is more loose what I had to present today.
Thank you, TJ. I had a quick question. In the beginning, you mentioned like a paper by Thomas, right?
Yes.
Is it Thomas Lu?
Garth Thomas. This was not or yeah, it's Garth Thomas. This was 2014. I believe he works in a lab at Shanghai or Chengdu. I can't remember which university he's at over there now.
Oh, okay, because it's interesting that you're talking about this global fMRI signal, and at
Mhm.
1 Eastern time in 25 minutes at Brain Space Initiative, we have this talk by Thomas Lu from UCSD, Resting
Mhm.
State Global Brain Activity, Controversies, Hypothesis, and Discovery. So
Yeah,
I wonder if your talks are coordinated.
we didn't coordinate it specifically, but yeah, Thomas is great. He I think he gave that similar talk at OHBM this last year. But yeah, him and Taylor work very close together. Work that I am not showing here from their lab is talking about how the global signal and there's and different implications to respiratory, to uh, cardiac pulsation, and a variety of other things. Taylor has a really nice paper categorizing and organizing what the pieces are of that signal as well, coming out of that lab. But yeah, this is about half of what the Keilholz Lab is doing. They're really what's being focused on right now is to do an awake animal model in the scanner, and that is very difficult to do, especially when you have exposed brain. But I know Wenju Pan has gotten very close, and they have the rodent on a treadmill able to be scanned right now, which is great.
That's interesting. By the way, we have 13 people here, so you don't have to raise your hand. Just pretend we're in the same room. I think it will work out. If you want to talk, just unmute yourself and
I'll ask a question that I've gotten asked before when I've talked to people about anything dynamic in fMRI. Do you think any of the dynamics in fMRI are actually real or all just noise?
No, they're
Yeah.
definitely real, especially because so a piece that I also didn't show here is Lisa Mayer. Shoot, I'm blanking on names right now, but Lisa's work is specifically looking at coordination of pupil diameter along with local field potentials and the Bold signal. And we are finding that these waves do coordinate even some of that information. So we're finding it not only in humans, in rodents, we're finding it in local field potentials, we're finding it in uh, dilation experiments with rodents. And so I do believe there is some coordination going on here. How we define that is going to depend on how more advanced we can make these methods and at least application of this data. So I really do think the work that Danju is working on, I'm working on, Harry Waters is working on, Lauren Daly's working on, we're all finding different aspects to apply this to, and hopefully it's a lower hanging signal as we continue to go on. This is nowhere near clinical ready yet, as much as MRI is not ready for clinical differentiation yet, but understanding how these networks sort of get coordinated will help us as we increase TR and acquisition time. And as
I
our
had a guy at
yeah.
OHBM just like talk to me for 5 minutes about how he thought dynamic analysis in fMRI was all pointless. And
Well,
so
I
I
mean, there
asked
was
that to
even a
everyone.
keynote. Yeah,
Yeah.
there's a keynote lady that said task was the only thing we should be doing and rest is pointless.
Exactly. But now that you mentioned, so I'm curious about, and you mentioned clinical ready, and that made me think of, stuff with EEG that's happening. I think one of the cool things that, some of these methods have as we, simultaneous EEG and fMRI becomes a bit more popular as a research method as well. Do you see any like pathway for doing like a joint cPCA in particular? Because there are a lot of complex base methods with EEG. I mean, I've worked on WPLI and stuff like that.
Yep.
You can, I think there's quite a cool way you could probably combine those two signals in an intelligent way using that, the cPCA method in particular.
Yeah, so there is one also research I didn't put here, I apologize, but Zhaodi Zhang, who graduated 2021, he ended up having a paper where he mapped EEG recordings and a Bold signal through a variational autoencoder, and the latent space actually was the QPP patterns. And so understanding the link between the two might actually give us a better encoding between. He did specify the window length, which might have given some bias there, but there is further research that we can do about that to see if there is a more of a latent space using some of these nonlinear neural net methods. But
So I have a question.
yeah, answer.
Sorry, I thought that
No,
was a
please.
spot. You mentioned that this were the animal model that they chose to do was were mice specifically. How transferable is that to human beings? And then why not chimpanzees? Just curious. And then
yeah, no, really great questions. Rodents are difficult to deal with, especially because a lot of times when you have to put them in the scanner, you have to sedate them. And so dexmedetomidine or isoflurane, or even a combination of the two, our lab has tried to demonstrate over the years of what is actually comparable to humans, as we need to have these animal models to be able to transfer to humans. And so one thing that our lab has shown is that a mix of isoflurane and dexmedetomidine, there's a happy balance between the two to have a very similar resting state that doesn't take away from functional connectivity. The reason that we do rats instead of mice is the brain is just that much bigger, so we can actually see some of these differences. Like here, we probably have, I think, 40 by 30 pixels across. With mice, you're going to get less, or even less signal to noise. Chimpanzees are now we're not ethically able to use chimpanzees. Specifically, I think 2 years ago, there was a new law passed that all chimpanzee research existing has to conclude, and then no more research will be handled, just because there's ethical concerns on them being locked up in such small habitats. And so PETA did that. I recommend the Crazy Chimp documentary that just came out by the Tiger King people. That'll put some light on like how terrible some of those cages are. However, we can use owl monkeys now and macaques. They have, I think, a third smaller brain size than chimpanzees, but we can potentially use that. We have not applied this to, or actually we have. Gosh, I think Garth also did a monkey study as well. We were able to do that and did find QPP patterns within the non-human primate data as well. But I'd have to go dig that paper up. But Jordan, long answer to a couple of questions, but does that sort of help?
that does. Yes, I think I remember that law. I'm still kind of bummed about the octopus, but thanks.
Yeah, no worries.
Just wanted to add here something. I saw a study that they had done on dogs, and they trained them to stay still in the fMRI machine, and
That's cool.
they were able. It wasn't actually a paper; it was part of a that shows 60 minutes, something like that. It was just a clip. So I mean, I didn't go into the technical stuff of it, but I did see that they have been doing that on dogs at least. Just wanted to add here.
So right now, Shella just got approved for like an exploratory grant where a master's student is going to look at the difference of Alzheimer's between human and rodents with these spatiotemporal patterns. And so I'm helping coordinate with that. So I think that's the not one of the first applications of cross-human or cross-species analysis, but at least within our lab. But hopefully, as we collect more data and get more collaborations, we'll be able to look at that. But yeah, it's always difficult to find exactly what we're looking for here when data is so kept under lock and key. It's a great questions. Am I missing anything in the chat? Let's see.
Oh, no, I was just adding a note. So I think Sajid mentioned about the dogs being scanned, and I was trying to add on that factor that professor is at Emory, Gregory
Cool.
Lawrence. Yeah.
Yeah, and I had mentioned earlier during the QPP stuff that one probably does suffer from the filtering stuff that Ashkan's looked into. Because if it does SWPC, I mean, it might not be a problem for your analysis, but it does, Schweidemann-Pearson correlation does filter your data, is the long and short of it. So you need to be aware of how it does that, yeah. And it's really dependent on the window size.
Okay, that would help.
Yeah, you might want to look at Ashkan's papers. I can send them to you because he has like quite a cool method for sort of modulating frequencies outside of the band that is affected by that filtering. And you can end up getting a much wider range of signal through a SWPC style analysis. Yeah, so I think it's quite cool, the stuff that he's working on.
Yeah, go and send those over to me, Brad. That'd be great. But yeah, I would say the other, maybe other applications to this, I know, I'm blanking on the name. There was someone in the Trend Center group who worked on a thing called statelets, which is very similar to trying to figure
That
out
was
some of these.
Muna's
Was that
work.
Muna? Okay.
Yeah, Muna based on stuff from Yosef, who I worked with yeah, back in the day. Yeah.
So that's a very similar method looking at some autoencoder latency. I haven't had a chance to run a similar analysis to this, but that is also trying to at least map out the spatiotemporal patterns within the data itself.
Yeah, Vince
Yeah.
has actually come across or come up with, I guess Vince and Armin are working on a method that's very similar to this template-based QPP stuff. And I wonder if they're aware of this.
Yeah, Armin's aware.
Okay, then yeah, they
him
probably
and I have been working on some stuff as well.
Yeah, yeah. So they're doing now a template-based DFNC essentially via spatially constrained ICA, and it ends up acting very similar to that QPP method in terms of deriving a template from some kind of SWPC-based method and then using it and apply it. It's fully data-driven because you derive the template from a huge data set, but it's quite similar in spirit, I think.
Mm-hmm. Yeah. So these large global propagating patterns do exist, and just trying to model it is going to be at least the conundrum that we'll have to figure out, as their recording devices are still pretty bad. Yeah, any other questions here? I know there's a lot going on here, so I apologize if it feels like a hose, drinking from a hose.
I have maybe I can ask just a sort of detailed question about rodent's frequency, this quasi-periodic, slides 8.
Sure.
You said milliseconds and 20 seconds, like fMRI in humans, hemodynamic response falls off about 19-20 seconds. But did you say, you mentioned some difference in frequencies of how often they go up and down for rodents
Yes.
and humans?
So,
Can you
so rodents they found that a window length of around 18 seconds, somewhere between 12 and 18 seconds was the best used for the template. This has more work with Machi Anumba. She ended up using QPPs a lot more, and Nanju, but they both used QPPs within rodents and found that having a window length of 18 to 20 seconds is better than humans, where we get the full waveform at around 20 to 30 seconds, with the best guess of 24 for human connectome project. But yes, with the rodents, it is faster. The best template was faster. And so we assume that with the smaller brains that there's going to be faster coordination between the networks.
That's the explanation.
Mm-hmm. How that can be tested maybe on a better gradient is, I probably should go look up the non-human primate paper, because I bet you there's a different window length for the template there too. What has not been fully confirmed or tested with is the window length with the complex principal component, or the cPCA. It's still around 30 seconds is the best, and we see the waveform start to repeat itself at around 25-ish seconds.
So
on average.
You call this global, meaning just that components or networks in different parts of the brain synchronize or interact. That's why you call it global. It's not like the word global.
So they are interacting. Do I have a better example here? Yeah, maybe here. So as we sort of go through, these would be the different components throughout the brain, and there is activity happening throughout, but it's a coordination of these signals over time throughout the brain. So that's why we're saying that it's more global.
Yeah, like, well, we all studying this brain networks, effective connectivity, functional connectivity for a long time, right? And I wonder, is it the same, what you mean global, that different ROIs or different networks in the ICA case interact, or you mean something else? I'm just trying the semantics of like...
Yeah, it is semantics. So really the global aspect here is that, yes, we're just talking about how the networks dynamically change over time. And so it's a coordination that we're seeing, or these waveforms are coordinating with each other that typically have these similar relationships of anti-correlation between the default mode and the task positive, or visual and somatomotor are usually coordinated and anti-correlated with the default mode network. And so these relationships are parsimonious throughout the brain over time.
So they are like the motifs, right? The way they
motifs, yes, we can call them motifs as well.
Okay, so it is like what Muna, similar to what Muna was looking at, and yeah. And there was a paper by Olaf Sporns, I think, or their team as well. They're looking at changes in the interaction, not changes in the activity, right?
Yeah, I think that was Faskowitz. They were looking at, I think, the weights between networks when they were constructing some of the graphs and sort of the motifs between those.
Yeah. That's interesting. I'm trying to find that paper. Complex brain networks graph. No. I'll find it later. Okay, yeah. So a lot of work and focused on that, and thanks for presenting a lot of
there's
an overview. Yeah, Brad, you were asking a lot of questions at this last slide. Did you get what's going on finally? Like I have a silly question, just why Hilbert transform, why not Fourier transform, or like I just that you said we'll go to details of the algorithms. So
I think there's a few things you could ask. There's like why not Fourier transform? There's like wavelet and like superlit based methods that could be really cool to do this with.
It's because the Hilbert transform specifically makes the data orthogonal within the phase, or the K space, I should say. And so we're able to have this 90-degree separation between phases. And that's the reason I believe we chose Hilbert transform, or they chose Hilbert transform over something else, or just like a agnostic frequency based one.
I think you could do that with wavelets as well.
Yes.
With Hilbert is simple. I get the part. It's like, why go to... Okay, I just didn't understand why Hilbert point. Like I mean, they're related, obviously. It was for
specifically, and I could maybe I can pull up the paper quickly. It was because it was applied already to climate data and something else. Shoot, I apologize. Where's the methods? Here we go. It was applied to atmospheric and climate science literature and engineering and physics literature as well. They call it a couple of different things, but this is essentially what they pulled it from. So it works with other things simply first.
Yeah, but I think there's a lot of ways you could kind of get into this space and do it, but the Hilbert transform makes sense as like an easy way to do it. I had another question, but have you looked into some of these other methods? So again, you see them more for EEG, but methods that do this nonlinear connectivity, quote unquote, often using information theory based stuff. So in EEG, there's WSMI, I think is what it's called, symbolic mutual information. And there's so the problem with fMRI typically is the temporal resolution is too low to get a lot of these metrics more reliable. But as we get higher resolution signals and higher Tesla coils, it's becoming more and more viable. And with weird sequences like EPTI as well, it becomes more viable. And then there are approaches that claim to get around it using copulas and other weird mathematical tricks that I don't 100% believe that they get around the sampling problem. Well, I don't believe they get around the sampling problem, but I think it's a cool approach. But have you looked into any of that nonlinear connectivity at all?
Not quite yet. I believe Amrit was starting to look into some of that. But it's been a minute since I've checked in with him. He's still over in Germany, and I don't think yeah, I don't think he's working quite for the hospital anymore, but it's been a minute since I've checked in with
Yeah,
him.
if you're interested in talking about it, I worked on it since UNM days, and we ran into the sampling problem. I dropped it. It got co-opted by a different student who ignored that problem. And then, there's like I have a whole sordid history with nonlinear connectivity, but I think it's really interesting and becoming more viable as we come up with new clever methods and higher resolution signals in particular.
Yeah, no, I'm super in. Brad, let's get a meeting on the books next week, and I'd love to talk to you.
I'm around. Yeah, let's do it.
Sweet.
Okay, thank you, TJ. Thank you everyone for listening. We have another talk. I posted the link in Zoom chat here if you want to connect. Brain Space Initiative in 3 minutes now. See you later. Bye-bye.
Thanks, everyone.